Thanks for giving me this opportunity. As Robert said, I'm going to give you a broad overview of what we have been doing during last few years on these type of pathogens. First of all, as you can see here in this slide, actually, these pathogens, they have very broad host range. You can call them all Fusarium diseases, but they can be somewhat different as well. But nevertheless, you can identify two major groups of pathogens, Fusarium pathogens that can cause damage to crop plants. You can have Fusarium oxysporum and related Fusarium species causing wilt disease in many important crop plants, such as bananas. I think Elizabeth will be covering actually in her presentation in coming weeks, I think, or next week. Also in cotton, Fusarium wilt, but also a number of other horticultural commodities like tomatoes, legumes, even in some tree species like palms. But also these pathogens, different Fusarium pathogens can cause also rots and blights and those kind of things in cereal crops mainly. As you can see here, for instance, this is a crown rot pathogen caused by crown rot disease caused by Fusarium pseudograminarum, and this is head blight disease caused by a related Fusarium species, Fusarium graminearum. But also there are other Fusarium species such as Fusarium verticilloides, which can cause great damage in maize ears. So the common, one of the common aspects of these Fusarium pathogens, I guess, they produce these mycotoxins. Some of these mycotoxins can be virulence factors that are needed to infect for the pathogen to infect its host. But also, while doing that, obviously, mycotoxins can accumulate in grains or in other crop plants that are consumed by people or animals. So they are quite, for that reason, important groups of pathogens. As I said, they can even cause disease in palm trees, actually. This is a picture taken, I have downloaded from internet, I think somewhere in San Francisco, causing this type of symptoms on palm trees. We can go even further, actually. Fusarium pathogens can also cause problems in animals. In this instance here, one of the Fusarium species, actually, Fusarium, I think it's Fusarium solani, infecting the eggs of these important turtle at Cape Verde, and it's threatening, actually, the species. And this is a story, actually, published in a scientific journal. So it's not I guess, newspaper news. But furthermore, certain Fusarium species actually can cause, as you can see here, rather grotesque infection symptoms on humans, especially in immunocompromised individuals. So overall, these groups of Fusarium fungi, they are quite obviously important pathogens for all sorts of lives, I guess. So the reason that I put actually this picture, there are some caves in France. Several years ago, people have located them, actually. You can go underground. In these caves, prehistoric humans, they have painted very nice pictures of the cave walls. This is a horse. When I was looking at this picture, actually, I thought the older horses, they were a little bit bigger. They had bigger bellies than the current horses, I guess. That's how they depicted in this picture. But the thing is that obviously this is a museum that you can visit. Here's the entrance. But what is happening at the moment, actually, in this place, they are having some problems from a fungal species that's infecting these beautiful paintings on the cave walls. And then it turned out that this species, actually, of fungus causing this pathogen is damaging effects on these paintings is a Fusarium solani. And it seems to be that this pathogen is extremely difficult to eradicate, because even using normal fungicides and bleach and those kind of things, obviously you don't want to damage the picture, but it was found that it forms a symbiosis, actually, with this bacterium called Pseudomonas fluorescens. And these bacteria can actively detoxify some of the fungicides that people have used to eradicate this fungus. So it turned out that it became extremely difficult to remove these fungi from these beautiful paintings. So overall, I guess these examples illustrate that I guess it is quite easy for these pathogens, these group of pathogens, to cause disease in a number of plant species. A couple of years ago, we became part of the Fusarium a consortium, actually, that has started sequencing of the genomes of these Fusarium pathogens. And the consortium actually was in the Broad Institute in the USA. We have contributed with some sequence data. But these are the major species of Fusarium, actually, that we are dealing with. Fusarium verticilloides, that's a maize pathogen. And then we have Fusarium oxysporum, causing wilt disease in a number of commodities. And we have this Fusarium gramineorum, that's the head blight, as I mentioned, and Fusarium solani, that's the potato and solanaceous infecting pathogen. So when you compare the genome sequences of these pathogens, Fusarium pseudogramineurum, as I said, is the head crown rot pathogen. If you look at the genomes of these, actually the Fusarium oxysporum infecting tomato seems to have, and other Fusarium species infecting Arabidopsis, they seem to have relatively larger genomes than some of the other Fusarium species. So one of the important things, actually, associated with these relatively larger genomes, these pathogens, obviously, they share a number of genetic material, especially these chromosomes. And we call them as the basal Fusarium genome. Most Fusarium species seems to have these basal genome with 4 to 11 largely synthetic chromosomes. But there are also individual genes located on these chromosomes, but they are Fusarium species specific, such as the red one that you can see there. But also on some chromosomes, you can find these gene clusters, like several genes in one chromosome clustered in one species, but in other species, you don't really see any of these clusters. So these are Fusarium species-specific regions. But also, interestingly, there are dispensable chromosomes or very strain-specific chromosomes, such as the ones depicted with red in this picture. They are specific to Fusarium oxysporum, for instance, in this case. And the other Fusarium species, like especially the tomato-infecting strain of Fusarium, contains these additional chromosomes. And I think one of the reasons why this particular species has larger genome is due to these Fusarium strain-specific chromosomes. But the importance of these chromosomes is that they contain certain genes that are important for virulence functions for the plant. So for some reason, during the evolution, these gene regions are accumulated on these chromosomes. One of the actually, I guess, interesting thing, sorry for the picture that's not that clear, but this is a good example that shows how important these strain-specific chromosomes are. This is a Fusarium strain, Fusarium oxysporum strains that infects tomato plants. As you can see here, they are all nearly dead. There is another species. This is, again, Fusarium oxysporum species, but this is not pathogenic to tomato plants. So as you can see here, plants are growing nicely. So if you isolate, okay, this is the strain name, actually, these two. If you isolate chromosomal components from this strain and then mix with that strain and then use that to inoculate the plants, as you can see here, this strain actually can become pathogenic. So when you look at the karyotype of this strain here, it was experimentally shown that they picked up some of these virulence-associated chromosomes, especially a chromosome called chromosome 14, that was sufficient by just incubating these chromosomal components with the strain to turn this non-pathogenic strain into a pathogenic one. So they can pick up easily genetic material from one another, and that can contribute to the virulence. That's a very important thing, how the virulence evolves in the field. One of the important things actually associated with these pathogens, they have different lifestyles. We normally classify fungal pathogens actually mainly into three groups. And some Fusarium species can fall into biotrophic group, like especially the one that infects Fusarium oxysporum that infects tomato. So just an overview of this type of interactions actually. So these are biotrophic pathogens, but they can, when they are infecting the plants actually, they release these molecules called effectors. And these effectors, they can be very diverse, can suppress plant defense responses. Obviously, when the plant senses the pathogen, it activates a defense response, as you can see here. But these effector molecules released by the invading pathogens have the ability to suppress these defense responses, and then you get normally susceptible interaction. You get disease. But if the plant has an R gene that has the ability to recognize some of these pathogen release molecules, then it triggers a stronger defense response as compared to weaker one here, and then you get resistance. So some Fusarium pathogens fall into that kind of biotrophs. They can act as biotrophs. But some of the others, they can be necrotrophs. So necrotrophs are just the opposite of biotrophs, like the crown rot pathogen that we talked earlier, and then this Fusarium head blight pathogen. They are considered normally necrotrophs. So necrotrophic pathogens, they release these cell wall degrading enzymes and a number of other toxins, as well as some effectors. And then even if plant has some defense, it's a constant battle actually between obviously plant defenses that can try to inhibit pathogen development, whereas pathogen try to overcome these plant defenses. So these necrotrophic pathogens, obviously, they cause necrosis, as the name suggests. As you can see here in this crown rot situation, for instance, that necrotic lesion at the base of the stem. And in the head blight, wheat heads are infected, and then as you can see here, the necrosis starts developing. As I mentioned, some of these pathogens, especially serial infecting Fusarium pathogens like Fusarium graminearum and Pseudograminearum, they produce certain toxins. And these toxins can be quite harmful if ingested by humans or animals, because they accumulate in the crop plant, in the grains, for instance. But also they have virulence functions. For instance, we found that this deoxynivalenol, that's a toxin, it's also called DON. That's a toxin produced by the Fusarium head blight pathogen, Fusarium graminearum, and also Fusarium pseudograminearum. So this pathogen, it causes actually, when you treat the plants with this toxin, as you can see here, it causes cell death. And the same as the fungus, actually, it causes similar cell death. So some of the effects that is fungus causing on the leaves, in this case, is mediated by DON. The effect actually behind that, as you can see here, if you apply DON at certain concentrations to plants, it causes this leathering, which is a typical hallmark of apoptosis. If you provide some ascorbic acid together with DON, that leathering is eliminated, indicating that reactive oxygen species associated with DON effect is causing that type of apoptosis or DNA leathering. So these type of I guess necrotic lesions on the leaves, they are quite important because they can be starting point of the infection. The infection can start developing from these dead cells. Necrotrophic pathogens, they love feeding on these kind of necrotic lesions. But also, some of the Fusarium species, some of the other Fusarium species, they can be called as hemibiotrophs. They are neither completely biotroph nor completely necrotroph, so they are somewhere in between, such as the one that Fusarium oxysporum species that we have been working for some time. This pathogen infects Arabidopsis plants. So this is a GFP expressing Fusarium oxysporum strain. It associates mainly with the root tips and lateral root initials. These are the entry points for the pathogen to get into the plants. And then, as you can see here, it starts invading the xylem, and then it moves up to the plant, and then it causes these typical lesions, or just starting, actually. In this case, the veins of the leaves are clearing, and then eventually it kills the plant completely. And then the spores can get into the soil again. As we know, Fusarium oxysporum spores can survive in the soil for long periods of time, like up to 30 years. And then when the susceptible plant comes along, it starts another infection cycle. So this initial infection cycle actually looks like a biotrophic type infection. It doesn't really cause any symptom up to 2 weeks. And then you get a necrotrophic stage where very strong necrosis develops. That's why it's called hemibiotroph. But these pathogens, as I mentioned, they also produce certain effectors. One of the relatively well-studied effectors is this effector called 6,4. 6 is a number of 6 effectors have been initially isolated from Fusarium oxysporum infecting tomato plants. So they are expressed mainly actually in the xylem of the tomato plants. When we have the genome sequence, actually, this is, again, as I said, it's quite conserved. We found it in Arabidopsis. I think Elizabeth's group has published some recent work saying that this is also in banana plants. This is the Arabidopsis 6,4 from our strain, this one. But it also shows there are at least 2 related ones in Arabidopsis, but one of them is showing very strong similarity to the one in tomato. So if you look at actually compare Arabidopsis 6,4 protein with the tomato one, there are only two changes. So it's extremely similar. So this 6,4 effector acts as an virulence gene actually in tomato plants. If the plant has a resistance gene that can recognize this effector, then it develops a hypersensitive response, and the plants become resistant. But in the situation we have in Arabidopsis, it's a little bit different from the one in tomato, because this interaction appears to be relatively quantitative. So we don't really have any one single resistance gene that can recognize the Fusarium oxysporum effector from Arabidopsis. So we have done some work to be able to characterize whether this 6,4 produced by the Arabidopsis infecting Fusarium oxysporum strain has any role in virulence. So one of the good things that actually you can have in these pathogens, you can transform the pathogen to knock out the genes that you want to study its function. So in this case, that's what we did. We knock out the gene encoding 6,4 effector from this Arabidopsis infecting strain, and then inoculated the plants, Arabidopsis plants, with the wild type, as you can see here. And that is these are the plants inoculated with the knockout mutant. You can see the difference, I think, between these two. Obviously, this effector contributes to the virulence in a quantitative manner, as I said. The difference is quite visible. You can also score the disease. You can show the differences. So there is a difference. When you complement, actually, this mutant with the wild type 6,4, you can also make it again more virulent on Arabidopsis plants, just like the wild type, indicating that this 6,4 effector has some role during infection in virulence. As I said, over the years, we have been studying this interaction between Fusarium oxysporum and Arabidopsis to understand what kind of mechanisms that this plant has, Arabidopsis, to protect itself from this pathogen. Obviously, there is no need to mention probably that the Arabidopsis is a model plant, and it has been widely studied, a number of mutants available, so it's easy to use in the lab to dissect some of the responses. By studying this interaction, we are finding that the hormone signaling pathways seem to be affecting the plant defense rather dramatically in this particular interaction. Especially to our surprise, especially jasmonic acid pathway. This is one of the important defense pathways that contributes to plant defense. It is also associated with plant defense in Arabidopsis against Fusarium, but it is a rather strange way that I will be going through. So basically, the SA pathway, that's another important pathway that seems to be contributing to the resistance of Arabidopsis to Fusarium oxysporum. But jasmonic acid and auxin signaling, they seem to be contributing to the susceptibility side. So the interaction between Arabidopsis and Fusarium seems to be like in a balance of different plant hormones. Depending on the direction, you get either susceptibility or resistance. As I said, we have been studying this Jasmonate pathway for some time as well. And when we started using this Fusarium oxysporum, again, we found that jasmonic acid signaling pathway actually provides susceptibility to the pathogen. And then after doing a number of experiments, we are now concluding that this pathogen can hijack this important plant defense pathway to cause disease. So let me just briefly introduce you to the Jasmonate pathway. As I said, this is an important plant defense pathway against pests and pathogens, to a number of ones, especially in particular to necrotrophic pathogens. So when the plants are attacked by fungus or insect pest, they produce this jasmonic acid active form, jasmonyl isoleucine. And then, obviously, this hormone is perceived by CoE1 JAS core receptors. So the CoE1 is an F-box protein that acts as a receptor for the plant hormone jasmonate. And then in more recent years, it was shown that CoE1 interacts with a number of proteins called JAS proteins. These are the proteins that are associated with suppressing plant defenses. So if there is no threat from any pest or pathogen, plant defense responses need to be suppressed. So these JAS proteins, they act as suppressors of Jasmonate pathway. But if the Jasmonate levels increase in the plant after a pathogen attack, this Jasmonate binds to these core receptor complexes, and then these JAS repressors, they are degraded by the proteasome. And then these actually JAS proteins, and normally they suppress some of the transcription factors like MYC2 and a number of others. So when JAS proteins are degraded, proteins like transcription factors like MYC2 can be released from the suppression, and then they go and interact with a number of downstream components, and then they bind to the G box in the promoter regions of defense genes, and they activate the defense. So this pathway, obviously important for producing defense-related proteins, but also it activates actually senescence symptoms as well. It's associated with senescence. And we think that this second aspect of the Jasmonate pathway is important for the infection mechanism used by Fusarium-oxysporum. So what seems to be happening actually, Fusarium-oxysporum also produces Jasmonates. It binds to these Jasmonates by binding to the COI1, activates this system, and also the senescence, chlorosis, and those kind of things. The reason why we are concluding this, as you can see here, these black arrows refers to the important proteins associated with this signaling pathway, the receptor, master regulator, and another downstream component that is important for transcription of defense genes. So when we have the mutations in this gene, and in this gene, we get all increased resistance in the plants. As you can see here, COI1 plants, they are completely resistant to the infection by this pathogen because they cannot sense any more pathogen signals. And then they don't activate senescence phenotypes that can contribute to the disease development. But importantly, another aspect of the, obviously, this pathway is the activation of defense-related genes. But their contribution to the stopping of the disease is much less than these ones, because this senescence, activation of senescence, that's something critical for this particular pathogen to cause disease. As I mentioned, you can see in this picture clearly that when we have the COI1 mutant, that's completely resistant, whereas wild type is developing these symptoms. So the idea behind this, as already mentioned, Fusarium-oxysporum by producing jasmonic acid, but also by activating both plants' jasmonic acid pathway, but also by producing jasmonates, activates these two downstream pathways. One of them can activate senescence, which positively contributes to the disease development, but also it has roles, of course, production of the antifungal defenses. They can negatively, they can suppress disease developments, but the effect of this pathway is much larger than this, so that you get increased disease development. So it's a kind of complex crosstalk between two different pathways. As I mentioned again, that's the mediator subunits that we have studied. The same thing is happening here. If you have mutation in this gene here, you get increased resistance, whereas if you overexpress this gene, you get increased susceptibility. This is one of the important mediators of the Jasmonate signaling pathway. It provides susceptibility again. So we seem to be getting a lot of susceptibility things. We have studied other mediator subunits. They seem to be also associated with disease, but without affecting actually the Jasmonate pathway. So there are different mechanisms of susceptibility that the plant has. When you knock out these susceptibility genes in the plant, as you can see here, in the case of MED18, for instance, you get complete resistance. Also, we have been studying where the resistance is happening. Like in this case, in MED18, we have done some grafting experiments to understand whether the resistance that we see, whether it's mediated in the roots or whether in the leaves. So if you graft COI1 on COI1 or wild type on wild type, as you can see here, you get disease symptoms. And MED18 mutant grafted on MED18 mutant is resistant. But when you have MED18 as scion, you get disease development. But if you have MED18 in the roots, as rootstock, you get increased disease resistance, indicating that MED18 functions, this mutant affecting this resistance, it seems to be happening in the roots. So these kind of grafting experiments that you can do in Arabidopsis seems to be extremely useful to understand whether the resistance is happening in the roots and in the shoots. Here is another picture used when we inoculated MED18 plants with gas-expressing Fusarium oxysporum. You can see that wild-type roots are being infected by this Fusarium oxysporum at much higher levels than MED18. Again, this provides another level of evidence indicating that the resistance may be happening in the roots. But also, in terms of this Arabidopsis pathogen, we are getting also development and disease. They seem to be cross-talking. In certain cases, the resistance phenotypes. In the resistant mutants, we often get a late flowering phenotype as well. As you can see here, like MED8 and MED25, two Fusarium oxysporum resistant mutants, they are both delayed in flowering responses as well. And we think that there is a link between flowering and plant defense against this pathogen, but I'm not going to go into details of that. So, because we have seen that a number of mutants that we have isolated, they seem to be associated with susceptibility rather than resistance, we decided to undertake a large-scale screen using thousands of available homozygous Arabidopsis tDNA insertion lines. And then we have inoculated large number of plants, incubated in our controlled environment rooms in level 4, and then started dissecting the responses. So again, what we have seen actually, through this system, we have isolated we identified a number of other mutants. When you, in the mutants, you see resistance phenotypes, indicating that wild-type genes are associated with resistance. Susceptibility. Like in this mutant here, one of the auxin mutants, you get complete resistance, whereas in the wild type, you get quite a bit susceptibility. So for some reason or another, resistance in against Fusarium oxysporum in Arabidopsis seem to be acting through susceptibility genes. More recently, we have done some RNA-seq experiments to further confirm that. The genes that you can see here in differentially expressed in roots and leaves of Arabidopsis plants, you can probably read all these, but all these genes that we have identified, they are one way or another associated with auxin responses, either auxin transport, auxin signaling, or auxin biosynthesis. They are all differentially expressed in Fusarium oxysporum infected roots. If you inoculate Fusarium oxysporum in these plates, you can see actually shortening of the roots of Arabidopsis plants, and you get increase in lateral root development, indicating that again, Fusarium oxysporum is changing, I think, certain processes associated with auxin signaling to develop these type of phenotypes. Again, we have done a number of auxin mutants using a number of auxin mutants, such as the one called his ARF2 mutant. When you knock out in the knockout plants, you get increased resistance. We have done tested a number of publicly available mutants in the auxin signaling pathway. As you can see here, all these arrows, when we have mutations in these genes, we get increased resistance, indicating that these wild-type genes, they provide susceptibility associated with all different auxin responses. So by doing this type of mutant analyses, we are trying to understand, trying to develop what determines resistance or susceptibility in Arabidopsis against this pathogen. So I'm not going to go into detail, but some of the things that I mentioned already, it's already placed on this picture. We are trying to, I guess, generate a larger picture of what seems to be happening during the interaction between Arabidopsis and the Fusarium oxysporum. But in the rest of my talk, I will be a little bit talking about the Fusarium pathogens that infect major cereals. Like as I mentioned before, this is the crown rot. It's quite chronic disease in Australia. It causes the Fusarium pseudograminiorum is the causative agent, but also we get head blight as well. It's a worldwide problem. It produces this toxin. It affects the quality of grains. These two pathogens, they cause quite a bit damage. Crown rot and head blight is quite widespread in Australia, especially around the, what we call is wheat belt of Australia, according to our surveys. In our region, actually, both head blight and crown rot can be seen, but in most other regions, mainly the crown rot caused by Fusarium pseudograminiorum. So we do quite a bit of work on these two pathogens as well. One of the things that we have done earlier is to sequence the Fusarium pseudograminiorum genome and then compare it with other genomes. Obviously, by doing genome sequencing, you can obtain quite a bit of information. One of the tools that we had in that time, actually, this is a couple of years ago, the genome of Fusarium graminiorum had been already sequenced using traditional sequencing techniques in 2007, I think. And because of the similarity between these two pathogens, obviously, our next generation sequencing data can be aligned to Fusarium graminiorum sequence to see how similar they are. The only 6% of the Fusarium pseudograminiorum genome could not be aligned into Fusarium graminiorum, indicating that this is specific to Fusarium graminiorum. But the rest, Fusarium pseudograminiorum, but the rest could be aligned quite well. You can see some gaps in this alignment. And when you look at closely these gaps, actually they, as you can see here, they are at the end of each chromosomes. And they are also associated with high SNP densities. One of the important things in these pathogens, they are telomeric regions of these chromosomes. They are quite variable, indicating that some of the virulence functions actually may be conferred by these, some of the virulence proteins may be located in these chromosomal ends. So when we looked at the Fusarium pseudograminiorum genome a little bit more closely, we were trying to identify Fusarium pseudograminiorum genes that may be shared by other cereal pathogens. Because we thought if we identify crown rot specific genes that are shared by other Fusarium pathogens, maybe we can, or other cereal infecting pathogens, we can try to identify commonalities between these group of pathogens, how they have adapted to their cereal hosts. This is an example of this. It's quite complicated, but these are Fusarium pseudograminiorum genes. And these are a number of other fungal pathogens, and these ones with blue color, they are grass pathogens. So, and then there are some other pathogens, they don't infect cereals. But what we are finding actually, a number of Fusarium pseudograminiorum genes, they have quite close blast matches with other cereal infecting pathogens. For instance, this Fusarium gene, Fusarium pseudograminiorum gene, it has a close homologue in Fusarium graminiorum, also in Fusarium oxysporum, but there is no other homologue of this gene in other fungi, even in cereal infecting fungi, but there is a close match in the bacteria. And we have identified a number of such genes. Like for this gene, there are quite significant matches to many other pathogens, including some cereal pathogens. For instance, in this case here, this is quite interesting case. This is the Fusarium pseudogramineurum gene that seems to be also present in Stegonospora nodorum that cause gulumbulage in wheat. That's another Fusarium, another cereal pathogen. No other sequence fungi contain this particular gene, but also very close matches in bacteria. So this gene encoding an amidohydrolase. So based on this type of analyses, we are trying to develop a hypothesis saying that maybe some of these type of genes actually may have been derived from bacteria through horizontal transmission. So this particular amidohydrolase gene actually, it also located at the end of the chromosome. As I was saying, these are the regions of innovation or virulence functions are developing by a number of means actually in this type of regions. So there is a cluster of genes in Fusarium pseudogramineurum that differs by, that is not present in Fusarium gramineurum. And this one here is the one that is encoding amidohydrolase. So we wanted to see whether this gene has really a function in virulence. So we have knocked out the genes in Fusarium pseudogramineurum and inoculated the plants using this system here. If you inoculate with the wild-type pathogen, you get obviously quite a bit disease. But these two mutants, they show reduced disease development, and this is MOC. So indicating that there is a function associated with this amidohydrolase in virulence. This is in barley, but in wheat you can see similar things. The reason I guess we are using these type of assays, given the virulence function conferred by these fungal genes is relatively small, you can really quantify very well by doing this type of experiments and then counting the survival. As I said, this gene only in Stegonospora nodorum and in also Fusarium pseudogramineurum, these two grass pathogens, they are separated by a large evolutionary distance. So, and there is no other sequenced genome that contains these type of genes. Again, indicating that they may have been transferred from bacterial organisms to these cereal pathogens. That is our model at the moment. So this amidohydrolase is probably from an unknown bacterial donor moved to Fusarium pseudogramineurum, but also Stegonospora nodorum. There are other examples of such gene exchanges actually between fungal organisms, like this toxin gene from Stegonospora nodorum moved to Phytophthora tirtici, causing tan spot quite recently. And some of our genes that we are finding in Fusarium pseudogramineurum, they are also shared by distantly related Fusarium and even not an even Fusarium species, indicating that there may be gene exchanges happening during evolution between these pathogens. So obviously, they are all contributing to the virulence. The last topic that I will briefly cover actually is the use of Brachypodium as a model for the Fusarium crown rod interaction. This work was undertaken by Jonathan Powell. He's a PhD student in coffee. Obviously, this plant is quite related to wheat, and due to obvious reasons, the genome has been sequenced completely, and there are a number of mutants available. It's a diploid plant. We thought we can start using this system as a model for Fusarium crown rod interaction at least. So we have, Jonathan has developed a very good infection system, and this is the scoring of Brachypodium seedlings after crown rod infection. As you can see here, some of the plants actually completely devastated, and then we have developed this scoring system from 0 to 5. Using this system, Jonathan has looked at a number of Brachypodium ecotypes available. Again, most of these are diploid species, as you can see here, and there are some polyploid species of this Brachypodium as well. And we are finding that these Brachypodium polyploid species, they seem to be more resistant for one reason or another to crown rot infection. Here are some pictures that shows Fusarium-infected Brachypodium plants. This is one of the ecotype, 2 different ecotypes. As you can see here, after the infection, the plant's completely dead. This is one of the polyploid accessions. It looks, as you can see, less disease. But also we are at the moment undertaking some experiments, RNA-seq experiments, to determine whether we can identify some processes associated with Fusarium infection in Brachypodium. This is infected versus mock. We have done some infection assays and then RNA-seq experiments. And also we have at the same time used wheat plants actually side by side to determine whether genes induced in Brachypodium after Fusarium crown rot infection can match to wheat genes induced by the same pathogen. As you can see here, we are getting really quite a bit good correlation between Fusarium genes that are induced after infection with Fusarium crown rot as compared to wheat genes. And there are some differences in induction between different genomes of wheat. We can determine those genes, but overall there is a strong correlation between genes induced by wheat and in wheat and Brachypodium by this pathogen. So this is one of the I guess picture that we are picking up some of the important pathways induced in Brachypodium after Fusarium crown rot infection. This is one of the phytoalexin biosynthetic pathway. All the genes associated with this pathway seems to be induced in Brachypodium, but there is, I guess at the moment, difficulty of identifying this compound. It's called BOA actually. That's one of the phytoalexins that is produced in wheat, in rye, but not in some of the other cereals. Genes encoding this phytoalexin seems to be induced by Fusarium infection, but we are yet to identify the actual compound, whether the actual compounds are also induced, given that there are differences between different cereal species in terms of producing this particular phytoalexin. But other indoor-related pathways that can give rise to a number of other defensive compounds, such as this the chorismite pathway that produces the SA. It seems to be also induced in Brachypodium, and we can, using metabolite analysis, we can also detect increased SA levels in the plants. So with this one, I think I will leave and thank a number of collaborators. Probably I have missed a number of names there, but overall, I guess we are trying to, using both models and crop plants, we are trying to understand how these pathogens can infect the plants, how these pathogens can change during evolution, potentially by gene exchanges, chromosomal transfers, but also how plants can defend themselves, and whether we can use that information in terms of improving resistance in the host against these pathogens. What I didn't cover in this talk, I guess, we have also pre-breeding activities in CSIRO. We are identifying QTLs associated with disease resistance in wheat and barley, and trying to incorporate these QTLs. If you had attended actually the the field day the other day, there were a number of talks around that area. Yeah, overall, our focus is on these group of pathogens. I think I will stop here. Thank you.